Hello everyone and welcome back to my YouTube channel. Today I'm here to teach you the last part of the chapter respiration in plants. So last time we were doing about some of the experiments regarding respiration and today we will continue with those experiments. So this is chapter 7. Okay, the name of the next experiment is prove that carbon dioxide is produced by green plants during respiration. We have all learned that what is process of respiration? Oxygen is taken in and carbon dioxide is given out. So we shall prove that experiment by way of an experiment. So what is the setup? Understand the setup, children. It consists of a bell jar. Okay, here you have a bell jar and you have a potted plant, a plant like geranium. Okay, and then you have placed this bell jar in between, and on either side you have two conical flask A and flask B. In flask A also lime water is taken, flask B also there is lime water. Now there is a inlet, okay, for the air to come in, and when at this inlet, at the beginning of the inlet, there is soda lime placed. The purpose of the soda lime is to absorb the carbon dioxide. Okay, so this air that is entering into this is free of carbon dioxide. So obviously the lime water will not turn milky and that is why it is clear lime water. Now this air that is free of carbon dioxide will pass through this tube and enter into the bell jar. Okay, so this air that enters into the bell jar does not contain carbon dioxide. Another thing that is done is this bell jar is wrapped with a black cloth. Okay, and this experiment is also performed during the night time or in a dark place. The reason for this is the carbon dioxide. Okay, the carbon dioxide that is given out during the respiration process will be otherwise used up for photosynthesis, because you know that during photosynthesis carbon dioxide is taken in and oxygen is given out. So they do not want the carbon dioxide to be used up. Okay, so that will not help us to prove the experiment. So that is why this bell jar is covered with a black cloth and the experiment is done in a dark place. So you can be asked the give reason why the bell jar is covered with a black cloth. Okay, so during the respiration, what will be happening is carbon dioxide will be given out by this plant, and obviously this air is going to pass through this and it is going to enter into this flask. And here what will happen? The lime water will obviously turn milky, hence proving the experiment, hence proving the aim of our experiment that carbon dioxide is produced by green plants during respiration. Okay, now here we have a few observations and conclusions, or rather the questions that can be asked in your exam. One is what I mentioned before. Why is it carried out in the dark and why it is covered with a black cloth? You can say so that photosynthesis does not take place. Otherwise, the carbon dioxide given out during respiration will be used up for the photosynthesis and will not able to help us to prove the aim of the experiment. Second question that is asked could be, why is soda lime placed, okay, at the entrance of the flask A? The reason is that carbon dioxide is absorbed, okay, from the air. That carbon dioxide is absorbed by the soda lime, so that the air entering the bell jar is free of carbon dioxide. Okay, now let's go to the next experiment. The next experiment is to prove that heat is evolved during respiration. You all know that during respiration, carbon dioxide is given out and also heat is produced. Okay, let's prove this with this experiment. So here you have two flasks, A and B. Now as you can see, it's like a thermos flask. It is insulating, made of insulating material, so that the heat cannot go in or be lost. Okay. So here you have in the setup A, you have the germinating seeds, and setup B, you have the dead seeds. How are they dead? Because you have boiled them. Okay, you boil the seeds and then you have to put a little bit of antiseptic into it. Okay, like carbolic acid, so as to prevent the decay of the seeds. Otherwise, there will be bacterial growth and the bacteria will respire and come in the way of your or interfere with your experiment. So that is why you are putting the carbolic acid. That is also a question that could be asked. Flask A, germinating seeds. Flask B, killed seeds with carbolic acid so as to prevent the decay of seeds. Now what you have to do, insert a thermometer in both the setups. As you can see in setup A, the thermometer reading has gone up. And in flask B, the thermometer reading is still down. The mercury level is still down. What does this prove? That there the temperature is much higher than this flask in flask B. Okay, so after a few hours, it is observed that flask A shows a rise in temperature while flask B, the temperature remains unchanged. And we also call this flask B the control experiment. Okay, now that also could be the question. Why is it called, which is a control experiment and why is it called the control experiment? Okay, so this is known as a control experiment because what is your aim? Your aim was to prove that heat is evolved. Now, always in the control experiment, remember students, the parameter will not be there that we are going to prove will be absent. Okay. Now, in this experiment, what was we trying to prove? Heat. Now, heat is absent in this. Heat is present in this. So, where it is absent, that becomes a control experiment. Okay, so in this case, control experiment is B because the parameter that is under study, that is heat, is absent in this experiment. So by this experiment, we have proved that heat is evolved only during respiration. Now we come to the next experiment, that is to demonstrate the anaerobic respiration. Okay, let's study anaerobic respiration. So here you have to take 8 to 10 seeds of pea which are soaked and peeled out. Okay, remember you have to peel out the seeds, peel out the seed coat. And what you have to do is push them into a test tube. This is a test tube, okay, in which you have pushed the seeds. These are the seeds of pea, and what is this black color thing is nothing but mercury. Okay, the shiny liquid metal at room temperature. Okay, and again down here also mercury is taken. Okay, it is inverted in a beaker containing mercury. Now what will happen? The seeds will float. Obviously they are lighter than mercury, so they will float on top, and but they will float, and here you have pushed, okay, enough mercury into the test tube. Outside also there is mercury, so the mercury level will go up here, and there will be, all these seeds will be covered with mercury. Leave the setup for few days, for 2 days, and the level of mercury falls down. Okay, after 2 days when you take this experiment, see, you can see this after 2 days, 2 to 3 days is written here. Okay, the level of the mercury will dip down. Okay, the level of the mercury dips down. And why is it so? Why is it so? Because carbon dioxide gas is given out. Okay, why? Because these species have undergone respiration, and during respiration, what have they given out? They have given out carbon dioxide gas, and that carbon dioxide gas has no place. Okay, so it has pushed the mercury level down. Now, how do you know that the space here contains carbon dioxide? Well, that can be proved by taking a stick of potassium hydroxide. You take a few pellets, okay, a few sticks of potassium hydroxide is introduced into the test tube. Now, potassium hydroxide will go up, okay, since it is lighter, it will move up, and potassium hydroxide has got the property of absorbing carbon dioxide. Okay, so it absorbs the carbon dioxide gas, and what will happen is, again, again this level of mercury will go up. Once all the carbon dioxide is absorbed by the pellets of potassium hydroxide, the level again goes up. Okay, so peeling off the seed. Now these are the questions that can be asked. Why is KOH used? Because it absorbs the carbon dioxide. Next one, why do you have to peel the seed coat? Okay, why you're removing the seed coat of the pea? Why not put it with the seed coat? Simple reason is that when you peel off the seed coat, okay, you're exposing more of the surface area, okay, and that will be increasing the rate of diffusion, okay, diffusion of carbon dioxide. So the carbon dioxide that is released by the seed, okay, during respiration will be able to be diffused into the air faster rather than if it is having the seed coat. Next question that could be asked regarding this experiment is, why is mercury used in this experiment? Okay, why is water not used? For the simple reason that water contains oxygen, okay, dissolved oxygen, which may hamper the experimental results. Okay, also why mercury is preferred, because you know mercury is a slippery metal which will not stick to the sides of the test tube also. Okay, so it will give a good result. then mercury does not react with carbon dioxide. Okay, so mercury, when the carbon dioxide is released, that carbon dioxide will never react with mercury. Okay, so this is also another reason. And obviously you all know that it is the only metal which is a liquid, which is in the liquid state at room temperature. So these are the reasons. Okay, so these reasons can be asked. You can be asked all this: why is KOH used, why is the seed coat peeled, and why is mercury used, okay, in this experiment. So these are the three most probable questions that could be asked regarding this experiment. Okay, now we have finished with the experiments. Now we go to the last part of the chapter. It is to differentiate between what is the difference between photosynthesis and respiration. Now photosynthesis, there is a presence of chlorophyll. You know that in green plants, all plants, they require chlorophyll. Okay, it takes place in the presence of the green pigment known as chlorophyll. While as respiration, it occurs in all the living cells of the body. Then you have occurs in the presence of light. Okay, now this it does not necessary that it will take place in the presence of light. This uses carbon dioxide and water. This will use oxygen and glucose. Oxygen is released as a byproduct. Okay, your carbon dioxide is released as an end product. Light energy is converted to chemical energy and it is stored in the body. Okay, so here it is light energy getting converted into that food. Okay, the starch. Your chemical energy is converted partly to heat. You know that during respiration heat is given out, and partly useful energy for various activities, and also a certain amount of energy is given out for our various acts or for the activities of the plant. Your weight is gained by the plant, your weight is lost. Food in the form of glucose is manufactured and your food is broken down. So here there's manufacture of food, you're broken down of food. So obviously, as you have learned, anything forming, anabolic process, breaking down, catabolic process. Okay, so these are the points you can be asked. Okay, related to maybe chlorophyll, maybe weight. Okay, you can be asked all such questions about the byproduct released. Now we come to the last part of the chapter, that is respiration in plants versus respiration in animals. Okay, well, in plants, the respiration, there is no gaseous transport. Okay, there is no gaseous transport from one part of the body to another part of the plant. Okay, like in animals, the blood is transporting the oxygen and the carbon dioxide. Okay, the oxygen to various parts of the body and carbon dioxide from the various parts of the body back to the heart. Okay, so that is a process that is going on. Okay, so that is a transportation that is going on. Next, the end product of anaerobic respiration is ethanol. Okay, in plants, you know, the anaerobic respiration, what is the byproduct is ethanol, whereas here it is lactic acid, which is the end product of your anaerobic respiration. These plants, they produce very little heat. Okay, plants produce very little heat during respiration, while as animals produce more heat during respiration. So that's all, and we have finished with the chapter. Thank you so much.